Hello dear friends, I'm excited to be back with you today. You can find only information about amino acids, peptides and proteins on my channel, which I have previously covered in 3 separate sections. We have comprehensive questions that I have compiled specifically for each section that cover the entire topic. Now get ready to discover the answers to these questions together while watching this video. Let's get started to the questions of first sections, amino acids, peptides and proteins 1. You can watch the video of explaining the section part 1 clicking on the upper right corner here. The first question is all proteins blah blah blah. So we have to fill the gap. Let's look at the choices. A. All proteins are synthesized using the same 20 amino acids. All proteins are synthesized from several amino acids. All proteins contain amino acids in the D form. All proteins contain amino acids in both D and F form. So I let you 5 seconds to think about the correct options. And the time is over, my friends. So remember, the building blocks of proteins are amino acids. And proteins in living organisms are usually composed of 20 different standard amino acids. Almost all of these amino acids are in the L form. D form amino acids are rarely found in protein structure and are usually found in the cell walls of some bacteria, or in some antibiotics, or in some different organic molecules. But for the proteins, L form. The correct answer is A, which the proteins are synthesized using the same 20 amino acids. And these standard amino acids are essential for the diversity and function of proteins and are determined by the universal genetic code. Answer is A. Which of the following amino acids is not chiral? Cysteine, glycine, glutamate. Remember, amino acids usually contain 4 different groups. Let me draw the groups. This is the general pattern of an amino acid. Remember it. An amino group attached to the carbon, and this attached to the carbonyl carbon, which makes an alpha carbon. This is an alpha amino group, and we have the R group. So to be a molecule, a chiral, there must be 4 different elements or 4 different groups. So X, Y, Z and A pattern. And so X, Y and A we have, but the rest is the R group here, which is the variable group, the variable carbon backbone. If the R is other than hydrogen, the amino acids are chiral. But if it is hydrogen or maybe another amino or carboxyl, so there will be no chiral carbon. And amino acids usually contain these 4 different groups attached to an alpha carbon. If all 4 groups attached to the alpha carbon are different from each other, the amino acid is chiral and can take the form of 2 different enantiomers, mirror images. All standard amino acids except glycine are chiral because the R group is different. Since the side chain of glycine consists of only 1 hydrogen atom. For the glycine, the R is hydrogen. And the 2 hydrogen atoms attached to the alpha carbon are identical, and therefore glycine is not a chiral amino acid, which makes the answer for this question option B, glycine. Let's fill the gap for the term zwitterion. Zwitterion is a positively charged phosphoderivatives of lipids, or zwitterion is the oxyform of carbohydrates. Zwitterion is called non-ionized compound, or zwitterion is a compound carrying both positive and negative charge. A zwitterion is a state in which a molecule carries both positively and negatively charged groups at the same time, usually in amphoteric compound. An amino acid exists in the zwitterion form at a certain pH, usually the physiological pH of 7.4. In this case, the amino group of amino acids carries a positive charge, while the carboxyl group carries a negative charge. And based on this information, the correct answer is option D. Zwitterion is a compound carrying both positive and negative charges. Amino acids as physiological pH, blah blah, are all alkaline, carry both positive and negative charges, are all acids, some carry negative, some positive charges. As you see, this is an alternate version of the previous question. Let me repeat. The physiological pH, such as 7.4, which is the normal blood pH, 7.4, actually 7.35 to 7.45. And at this pH, amino acids are in the Zwitterian form, meaning that they carry both positively and negatively charged groups at the same time. The amino group or amino acids gains a positive charge. So let me draw to show you. This is the Zwitterian form, and we have the R. And as you see, the amino group or amino acids gains a positive charge, is protonated by being this group, NH3, while the carboxyl group loses a proton, gains a negative charge. So based on this information, the answer is B. Carry both positive and negative charges. Which of the following is an amino acid containing an S? So what is S, my dear friends? It is sulfur. It's an element. So we are looking for an amino acid carrying sulfur in its structure. There are two amino acids carrying sulfur. They are methionine and cysteine. Look at the options. We have the methionine. Thus, the answer is B. Threonine is an amino acid carrying an alcoholic group. Tyrosine is an amino acid carrying an aromatic group, actually the phenolic group. And valine is an amino acid having a side chain. It's a branch chain amino acid. Which of the following is an amino acid containing an aromatic group? So you're lucky. You gave the answer in the previous slide. And as you see, we have the tyrosine. The options is same, but the question address a different answer. It's tyrosine. Methionine, sulfur carrying an alkyl form, and threonine is an alcoholic amino acid, and tyrosine having a benzene ring, and actually it's phenolic ring, and hydroxybenzene, and valine is ABCAA. Also, the question could be, which of the following is an amino acid containing a branch side chain? Thus, the answer would be valine. So please carefully read the questions. What is asking to you? GABA. What is GABA? It's an essential amino acid in cats. It's a non-protein amino acid that acts as a neurotransmitter. It's an important intermediate in methionine metabolism. It's a non-protein amino acid involved in the urea cycle. GABA. This is an abbreviation of, let me write it for you, gamma aminobutyric acid. And the GABA is a neurotransmitter and an amino acid that is not included in the protein structure. It's a non-protein amino acid, but due to being a neurotransmitter, it's a critical amino acid. It's a functional non-protein amino acid. And GABA acts as an inhibitory neurotransmitter, especially in the central nervous system. So based on this information, the option B is the best choice for us. This is the answer. It's a non-protein amino acid that acts as a neurotransmitter, and it's an inhibitory effect on central nervous system. It is found mainly in the substantia nigra, globus pallidus, and hypothalamus. And alcohol, barbiturates, and benzodiazepines increases the effect of gamma aminobutyric acid, especially in postsynaptic sites. Yes, sorry about the quality of the picture, but I think it's enough to answer the question. So there is a mark showing a chemical bond, and the question asks you, mark bond is which type of bonding? So you have 5 seconds. And yes, I hear that you say the correct answer. The mark bond is an peptide bond. From the structure of the molecule and the nature of the mark bond, I can tell that this bond is a peptide bond between two amino acids. As you see, these are amino acids. The carboxyl group forms an amide here. They have an amino group, a carboxyl group, and the nitrogenous side. So this means that there is a removal of water between two amino acids, forming an acid-amide bond. That's also option A is correct here. But due to the nature of acid-amide bond, which I mean the acid-amide bond occurred between two amino acids. Specifically, it is called a peptide. In this question, we have 2 correct options. The question about non-essential amino acids. They are not components of tissue proteins. It can be synthesized in the organism through essential amino acids. There are no roles in metabolism. They are synthesized in disease states. So choose the correct statement about the non-essential amino acids. You have 5 seconds. Yes, let's analyze each option one by one. They are not components of tissue proteins. This is incorrect because they are components of tissue proteins. So let me give an example for non-essential amino acid. For example, alanine. A non-essential amino acid. Alanine is a component of tissue protein. It can be synthesized in the organism through essential amino acids. And for some, yes, this can be accepted as a true option. For example, tyrosine. Tyrosine is a non-essential amino acid. But to synthesize tyrosine, we need the phenylalanine, which is an essential amino acid, which makes the option correct. There are no rules on metabolism. This is a completely, in parentheses, in brackets, I can say a ridiculous statement, completely incorrect. They are synthesized in disease state. Maybe or may not be synthesized, or it may not relate with the disease state. But in general, we can also accept this is a correct option. But the B is more correct. Collagen. We are looking for the correct statement. It's a connective tissue protein rich in homoserine. It is the main protein of skeletal muscle tissue and is rich in branched-chain amino acids. It is rich in cysteine. It is rich in hydrophobic proline amino acid. Think about the question. 5 seconds. It is time to answer. There are statements here explaining the structure of collagen, and it is desirable to choose the right one. Collagen is the most abundant protein in the body, especially rich in the amino acids proline and glycine. It is also known for its hydroxyproline content, a modified amino acid of proline. So among the options, the option D, it is rich in hydrophobic proline amino acid, correctly describes a characteristic feature of collagen. The answer is option. And the amino acids proline and hydroxyproline play a major role in the formation of the triple helix structure of collagen. Thus, the other options do not accurately reflect the structural properties of collagen. Therefore, the correct answer is option D. Desmosine. We have 4 options. It consists of oxidation of 4 lysine molecules and takes place in the structure of elastin. It consists of the oxidation of 4 proline molecules and takes place in the structure of elastin. It consists of the oxidation of 4 lysine molecules and takes place in the structure of collagen. It consists of the oxidation of 4 proline molecules and takes place in the collagen structure. So very similar options, and be careful and analyze them, all of them. So in option A, as you see, is for lysine, and also in this C for lysine, and statement tells us oxidation, they are same, but the protein is different, elastin and collagen. In option B, again, oxidation of 4-proline, so different amino acid, and same in the D. And again, we have elastin and collagen. So 4 different statements addressing different amino acids and proteins. Desmosine is formed by the oxidation of lysine amino acid, and it is found in the structure of elastin protein. Elastin is a protein found in connective tissue and provides elasticity to tissues. Desmosine plays an important role in providing elasticity by forming cross-links of elastin protein. So looking at the options here, the correct answer is A. It consists of oxidation of 4 lysine molecules and takes place in the structure of elastin. This option correctly describes the chemical nature of desmosine and the protein structure it is involved in. Which of the following amino acid, the AA abbreviation of amino acid, provides the chelation of calcium in the prothrombin structure? And you have the options beta-alanine, gamma-amino butyric acid, GABA, phosphorthreonine, and gamma-carboxyglutamate. So there, some are non-protein amino acids, and here some are modified amino acids. And gamma-amino butyric acid and the beta-alanine, they are the non-protein amino acids. Phosphorthreonine and gamma-carboxyglutamate are modified amino acids. Beta-alanine is building component of pantothenic acid, my dear friends, which is the vitamin B5. Vitamin B5, and it's a part of this coenzyme is the coenzyme A, very important coenzyme that we will study in metabolism. And phosphorylation of the hydroxyl groups in the side chain of the amino acid threonine or tyrosine and serine And what are the common features of these amino acids? All carry an alcoholic group. So the phosphate group is a stratified with the alcoholic group of this amino acid, providing the phosphoproteins. And these three rare amino acids, which phosphorylane, phosphotyrosine, and phosphoserine, are involved in the structure of regulatory proteins. Also in some other structures, such as lipids, phosphoserine, remember. And we talk about the gamma aminobutyric acid in the previous slides. So we have the option D remained as a true option. And the amino acid that enables the chelation, the binding of calcium in the prothrombin structure is gamma carboxyglutamate. Its abbreviation is GLA. And this specifically modified amino acid is formed by vitamin K dependent reaction and plays an important role in the blood clotting process by interacting with calcium ions. The correct answer among the options in the question is gamma carboxyglutamate, option D. Another question, we have a statement. It is a vasodilator compound that is produced by the decarboxylation of what? So decarboxylation is a removal of carbon dioxide group from an amino acid. So the question asks us to find which amino acid decarboxylate to yield a vasodilator compound. And if you look at the option, we have tryptamine, lysine, histidine, and histamine. As we know, histamine is a vasodilator compound, but it's not the answer because we are looking for the amino acid which yields histamine. And the answer is histidine. Histamine, just change the it to histamine. Histamine is a vasodilator compound that is produced by the decarboxylation of histidine. The enzyme is histidine decarboxylate, catalyzes the conversion of histidine to histamine. And histamine is involved in functions in the body such as allergic reactions and acid secretion, and increases blood flow by dilating blood vessels. And the decarboxylation product of lysine is cadaverine, and tryptamine is a decarboxylation product of tryptophan. What for the question? The answer is histidine. Be careful about here. Both options of histamine and histidine may create confusion for you. But the asking is about the amino acid, not its product, and not its product after decarboxylation. Be careful. We have finished the questions for our section 1, and it's time for the questions of section 2. And you can watch the video explaining this section by clicking on the upper right corner here. Which of the following is a tripeptide that acts as a reducing agent in tissues? And we have the ophthalmate, anserine, oxytocin, and glutathione. And ophthalmate, it's a tripeptide found in eye, the lens of eye. Anserine is a dipeptide and we found in the skeletal muscle and brain of mammals and birds. Oxytocin is a nonopeptide and it's a hormone synthesized by hypothalamus stored in posterior pituitary gland. And glutathione is a tripeptide, acts as a reducing agent in tissues. This is the answer. And glutathione is an important antioxidant that reduces oxidative stress in cells and neutralizes toxic compounds. It's a tripeptide. It's glutamyl, cysteine, glycine. So among the options, the glutathione is consistent with the explanation. So this is the answer. Which of the following is a metalloprotein? And you have the options glutathione, lipoprotein, ceruloplasmin, and gelatin. You have 5 seconds. Metalloproteins are proteins that contain one or more metal ions as structural components. And among the options given in the question, the only option that contains metal ions and is an example of a metalloprotein is ceruloplasmin. This is the answer, ceruloplasmin. Also, we did ceruloplasmin. We talk about ceruloplasmin in the bioelements topic. And ceruloplasmin is a protein that binds and transports copper ions and has a particularly important role in the transport and metabolism of copper in the body. Which of the following is a chromoprotein? And you have the options gliadin, myoglobin, protamine, and ferritin. The answer is myoglobin. Let me explain. Chromoproteins are a type of proteins that have a pigmented prosthetic group, such as haemoglobin with its red heme group. So they carry porphyrin complex, especially porphyrin complexes, carrying an iron. And this structure is called heme. And heme-carrying proteins are chromoproteins. The heme is the prosthetic group. So the myoglobin is a heme-containing protein. And in the structure, there is an iron, which makes it also a metalloprotein. What about the ferritin, gliadin, and the protamine? The protamine and gliadin they are the simple proteins. They only consist of amino acids. But ferritin is a metalloprotein carrying an iron, but not in the heme form. And yes, ferritin is important in iron metabolism. It stores iron and releases it in a controlled fashion. And the iron in the ferritin is a storage form of iron in the body, but it's a non-heme protein. Thus, it's not a chromoprotein. For mammals, especially for mammals, they might be different. But also there are heme-containing ferritin in bacteria ferritin. But here, for the context of the question, the answer is myoglobin. Blah blah is a simple protein found in the structure of nucleoproteins, which means they are the component of nucleic acids, or nucleic acids, and that proteins forms the nucleoproteins. And they are simple proteins, which composed of only amino acids. And you have the options glutenins, prolamines, protamines, and albumins. And I hear you said you are looking for the histones in the options, aren't they? And yes, histones are the main proteins in the structure of nucleoproteins and play a role in DNA packaging. However, this term is not included in the option. Histones are essential proteins in the structure of nucleoproteins and are not among these options. Therefore, the question may be incorrect, or we should look for another answer. And yes, we have another answer, and it is protamines. And protamines are also simple proteins found in sperm cells that play a role in DNA packaging. Which of the following is rich in sulfur, as S, which means rich in methionine or cysteine, and is found in hair and oral structure? An easy question. Elastin, collagen, fibrinogen, and keratin. So what is your answer? And yes, it's keratin. You are correct. Keratin is the sulfur-rich protein, cysteine-rich protein, found in the structure of hair and horn. It is the main structural component of hair, nails, horns, and rich in the amino acid cysteine, which contains sulfur to form crosslinks that give these tissues their strength. The cysteines form disulfide bridges. So the answer is D. Which protein is dissolved in blood plasma and is involved in coagulation, blood clotting? Elastin, histidine, fibrinogen, and myosin. Yes, the elastin is a connective tissue protein which provides elasticity and rich in blood vessels. Histidine is not a protein. Eliminate histidine. It's an amino acid. I'm asking a protein. And fibrinogen is the answer, my friends. Fibrinogen is a protein that is dissolved in blood plasma and is involved in coagulation. During the clotting process, fibrinogen is activated by the enzyme thrombin and converted into fibrin monomers, which then form crosslinks to form a solid clot. Therefore, the correct answer is C. And myosin is a protein found in muscle tissue. Lipase is an example of which type of protein? Transport, catalytic, contractile, or immunoglobulin. And lipase is an enzyme. It's an enzyme that breaks down fats, so it's a protein with catalytic activity. And catalytic proteins are enzymes that accelerate biological reactions. Lipase is therefore an example of category of catalytic proteins. So the answer is B. It's not a contractile, not a transport, and not an immunoglobulins. An example for immunoglobulins, for example, IgG for the contractile, myosin, and for the transport, we can give example such as transferrin or lipoprotein. The answer is lipase is a catalytic protein. Histones are protein rich in which amino acid? Arginine, alanine, valine, or tyrosine. In the previous slides, we talked about the histone. We had a question about nucleoprotein. And histones are particularly rich in the basic amino acids. And so, what are the basic amino acids? Arginine, lysine, and histidine. Especially histones are rich in arginine and lysine as a basic amino acid. And these amino acids contribute to the positively charged characteristics of histones, which allow them to bind tightly to DNA. And arginine and lysine bind tightly to the negatively charged phosphate backbone of DNA, strengthening the interactions between DNA and histones. And these interactions play an important role in the regulation of chromatin structure and control of gene expression. And so, look the options for the arginine and lysine, and we have the arginine. So the answer is A. Denaturation of proteins. It is the deterioration of its natural structure and is mainly happened by the breakdown of hydrogen bonds. It is the breakdown of the primary structure. It is the breaking of peptide bonds and the disruption of the tertiary structure. It refers to the irreversible reactions in the molecule. So what is the correct statement for denaturation of proteins? 5. Denaturation of proteins is the disruption of the three-dimensional structure of the protein, and in the process, the weakening or disappearance of the forces that stabilize hydrogen bonds, hydrophobic interactions, and other secondary, tertiary, and quaternary structures that usually maintain the protein's native structure. However, this process usually does not imply the breakage of peptide. So it's not a disruption of the primary structure. Of the options given, the option A, it is the deterioration of its natural structure and is mainly happened by the breakdown of hydrogen bonds, most accurately reflects the definition of protein denaturation. And during this process, the functional conformation of the protein changes, but the amino acid sequence, the primary structure, is usually unaffected. So the answer is A. It's not the breakdown of primary, or not the breaking of peptide bonds. And there may be reversible reactions, which is called renaturation, but often not happens, but may. The tertiary structure of proteins. It is the order of amino acids. It is determined by the position of the disulfide bonds. It refers to the regions where proteins form loops. It is determined by the protein folding pathways. The tertiary structure of protein is the three-dimensional folding of the chain of amino acids. The structure includes both non-covalent interactions such as hydrogen bonds, hydrophobic interactions, ionic bonds, and van der Waals forces, and covalent bonds such as the sulfide bonds, especially in the extracellular matrix proteins. The tertiary structure determines the functional conformation of the protein and is critical for its biological activity. Among the options given in the question, the B option, it is determined by the position of the disulfide bonds, reflects an important feature of the tertiary structure, especially in extracellular matrix proteins, because the sulfide bonds play an important role in the stability of the protein. And a good example is insulin, for example, which carry two polypeptide chain and they are attached with the disulfide ring. However, tertiary structure is not limited to the disulfide bonds. It encompasses the overall folding pattern in all interactions that stabilize this folding. So in this question, the option D, which it is determined by the protein folding pathways, also provides a general description of the tertiary structure of the protein and is the most comprehensive answer that describes how the three-dimensional structure of the protein is formed. Therefore, the correct answer is option D. But also we can accept B as a correct option for some certain cases. So we can say the question is the question has two correct answers. Don't forget, the best option is determined by the protein folding pathways. This is the primary statement. This is the secondary statement. Alpha helix and beta sheet structures in proteins are examples of what? A very basic question. Alpha helix and beta sheet structures in proteins are examples of the secondary structure of a protein. Secondary structure. And these structures are formed by folding amino acid chains in specific ways and are stabilized by hydrogen bonds. It's time for the questions of section 3, amino acids, peptides, and proteins 3. And don't forget, you can watch the video explaining this section by clicking on the upper right corner here. A nucleoside consists of what? A nitrogenous base, purine or pyrimidine base plus sugar, purine or pyrimidine base plus phosphorus, purine plus pyrimidine base plus sugar plus phosphorus. And the nucleoside consists of a purine or pyrimidine base and the sugar molecule, which makes the answer B. The sugar can be ribose or deoxyribose. And purine and pyrimidine bases are the nitrogenous bases. And if instead of plus we have or here, it gives a definition of a nucleotide. What are the purine bases? Adenine, guanine, the pyrimidine, thymine, guanine, and cytosine. A purine nucleotide, look at the options, AMP, CMP, UMP, and TMP. All are monophosphates, all purine or pyrimidine bases. They are the nucleotides. This is the common feature of all options. But we are looking for the one carrying purine instead of pyrimidine. And the answer is A, because the A refers adenine. It's a purine nucleotide. The rest, here the cytosine, uracil, and thymine these are pyrimidine nucleotides. The most abundant free nucleotide in mammalian cells is FAD, GTP, ATP, and NAD. And be careful, all are free nucleotides, but we are looking for the most abundant one. It's the C. It is ATP. Adenosine triphosphate is the most abundant mammalian cells and plays a fundamental role in energy transfer. ATP stores and provides the energy needed for cellular function. The nucleic acid base found in mRNA, but not in DNA. So we are looking for a nucleobase or a nitrogenous base, which is a part of mRNA, but not DNA. Very easy. It's uracil. In DNA, there is a thymine instead of uracil. So adenine, cytosine, and guanine, these are common bases found in both DNA and RNA. But uracil, not common. It's in RNA, not DNA. The structure of transfer RNA, tRNA, appears like a helix, hairpin, coil, or cloverleaf. And the answer is cloverleaf. Its molecular structure is often described as the L shape or cloverleaf. The sugar moiety present in RNA, which gives the name R here, represents the ribose, so ribose nucleic acid. In DNA, deoxyribose. Glucose is the oxy sugar. It's another sugar in RNA. Ribulose is another sugar. It's a ketopentose. It's a component of photosynthesis, not the RNA. tRNA, transfer RNA, contains what? Codon, anticodon, password, or anti-key locker. The tRNA molecule contains the anticodon region. This region has a triple base sequence complementary to the codons of messenger RNA and ensures the amino acids are added to the polypeptide chain in the correct order during protein synthesis, during translation. But the correct answer is anticodon. For the password and anti-key locker, they are not terms used in biology or genetics. Genetic information flows from DNA to mRNA, DNA to ribosomal RNA, RNA to cellular proteins, DNA to cellular proteins. The flow of genetic information is often expressed as DNA to RNA and RNA to protein. This is known as the central dogma and describes how genetic information is processed and translated into cellular functions. In this process, the flow from DNA to messenger RNA is transcription. So the option A tells us the transcription, and flow from messenger RNA to proteins, as in the C, for example, RNA to cellular proteins, describes the translation. So based on these informations, the option A and C correctly represents the two stages of the flow of genetic information. However, in option D, DNA to cellular proteins, which expresses the entire flow of genetic information, provides a general summary of this process and is the most appropriate answer in this context. So the answer is D, my friends. AUG, adenine uracil guanine, the only identified codon for methionine, is important as what? A releasing factor for peptide chains, a chain terminating codon, recognition site on tRNA, a chain initiating codon. So what would your answer? Think for 5 seconds. AUG, the only codon defined for methionine, determines the start of the chain during protein synthesis. It's a chain initiating codon. codon? The answer is D, my friends. So this codon is not a releasing factor and the terminating codon. The terminating codons are uracil, adenine, UAA, UAG, and UGA, which they do not encode an amino acid. They are stop codons, in other words. And the recognitions on tRNA is called anticodon. That's another definition, not the answer. The answer is D. Haemoglobin has a high content of which amino acid? Proline, lysine, histidine, arginine. And hemoglobin is the oxygen carrier. It has also buffering capacity. It contains 4 hemes, 4 subunits, 2 alpha, 2 beta. Each contains 1 heme unit. That's iron, and iron carries oxygen. So, what is the answer? And don't forget, the all given amino acids are component of hemoglobin, but I'm asking about the high content. And yes, you are true. The answer is histidine, and histidine is a critical function. Histidine is one of the amino acids that binds to the heme group of the hemoglobin molecule and plays a critical role in the transport of oxygen during this binding. So the correct answer is histidine. Arginine, yes, it's a part of hemoglobin, but does not play as important role as histidine. Lysine also found in hemoglobin, but not the abundant one, not the most abundant one. Proline also present in hemoglobin, but is not particularly abundant or plays a critical role as histidine. So the answer is histidine. Prehepatic icterus. So we will look for the correct statement describing the prehepatic icterus. It is also called as hemolytic jaundice, and there are increased rate of hemolysis. The hemolysis means destruction of cell membranes of erythrocytes. It's the lysis of erythrocytes, and jaundice is icterus. Icterus is a medical term. It is also called as hepatocellular jaundice, and there are cellular necrosis that decrease liver's ability to metabolize bilirubin caused by the obstruction of bile ducts, and it's a type of hyperimmunoglobulinemia seen in liver diseases. So think for 10 seconds. Icterus or jaundice. It's a state of hyperbilirubinemia, which means increased levels of bilirubin in blood. And there are 3 types of icterus, which we classified them according to the root cause: prehepatic, hepatic, and posthepatic. And here the asking one is prehepatic. And prehepatic icterus is usually characterized by an increased breakdown of the blood as a result of hemolysis, which increases the amount of hemoglobin released into the blood and thus increases the bilirubin level. And this is explained by the option A, which is the true option. Prehepatic icterus, it is also called as hemolytic jaundice, and there are increased rate of hemolysis. What about the other one? In the B, it defines the hepatocellular jaundice. It is defined as the hepatic icterus, a type of jaundice caused by damage to liver cells, and is hepatic icterus, not the pre. Pre means before liver. So there is an ongoing process. There is a pathway which bilirubin follows. It is released in the reticuloendothelial system, then transferred to the liver, and then released into intestinal tract. So we put the liver as a leading role in regulating or managing the bilirubin metabolism. There is something happen before the liver, we call it prehepatic. If there is something happens in the liver, it is called hepatic. And after the liver, we call it posthepatic icterus. What about option C? Caused by the obstruction of bile ducts, this is posthepatic icterus. Posthepatic icterus. And what about D? A type of hyperimmunoglobulinemia seen in liver diseases is not directly related to icterus. So this is not a statement about the icterus. Option A is the correct statement for prehepatic icterus. Serum does not contain what? Glucose, fibrinogen, albumin, and ceruloplasmin. What will your answer? Yes, you are correct. The answer is fibrinogen. Serum is blood-free of clotting factors and therefore does not contain fibrinogen because fibrinogen is converted into fibrin during clotting and becomes part of the clot. The serum does not contain fibrinogen. Therefore, the correct answer is B. Glucose is a component of serum. The sugar that we measure. Albumin is a protein found in serum, it is the major plasma protein, and ceruloplasmin also found in the protein, which has function in copper metabolism as well as in iron metabolism. And this is the final question of this video. Bilirubin is conjugated with what to be removed in the bile, and this is an essential process done by hepatobiliary cell, or shortly liver. Taurine, glycolic acid, glycine, and gluconic acid. What is your answer, my friend? Yes, well done. You are correct. It is glycolic acid. Bilirubin is conjugated with glycolic acid or short glucuronate to yield the bilirubin glucuronate in the liver in the hepatobiliary cells to facilitate its excretion through bile. This process makes bilirubin more water soluble, thus facilitates its extraction from urine and feces. And the correct answer is B. What about the other? Taurine and glycine. They are the amino acids, and taurine, don't forget, it's a non-protein, but glycine is a protein amino acid. They form conjugates with bile acid, not the bilirubin. And gluconic acid is not relevant with the bilirubin or as well as in bile acid, but gluconic acid is then aldonic acid of glucose. By the way, gluconic acid is the uronic acid of glucose, and they are both also components of some metabolic pathways that we will discuss in biochemistry too. And we finish. More about biochemistry, clinical biochemistry, and laboratory world, you can visit my website, BiyokimyaVET. You can also follow my Instagram account. Don't forget to subscribe to the channel, like the video, and turn on notifications to get updates. And thank you, my friends, and I wish you success. Goodbye, stay safe.